Welcome back, friends. Welcome to another video tutorial from Shromu's Biology. And in this video lecture, I'll be talking about the difference between transformation, transduction and conjugation. All these three techniques are the way to transfer genetic material from one bacteria to the other bacteria of the same generation. So it is also known as horizontal gene transfer or lateral gene transfer. So let's start to talk about the differences between all these mode of gene transfer. So first let me take a color. Let's take it. Now in all these cases, if I just put it in a simplified fashion, all of them are gene exchange methods, okay, between two bacteria of the same generation. Now the process of transformation where the DNA or the fragment of the DNA is taken directly by the adjacent bacteria. In transduction, the DNA or generally the genetic material was DNA, so the DNA is transferred by a bacteriophage virus from one bacteria to the other bacteria, which is this one. And the third one, conjugation, is when the genetic transfer occurs by direct contact between the bacteria. Okay, these are the three different situations that we know. Now in all this type of gene transfer, there are two types of organisms involved with. One is the donor, another one is the recipient. Donor and recipient. Donor provides the gene, recipient receives the gene. In all these three cases, this is true. Now let's talk about the difference in the major scale. First is the transformation, which is the uptake of naked DNA fragment by the bacteria. So this is the example. Normally what happens when the donor cell start to die, DNA fragmentation start to occur inside the bacterial cell. On the verge of apoptosis or the cell death, all those DNA fragments start to generate, the chromosomal DNA of bacteria start to be degraded, so as the cell wall, cell membrane ruptured of the bacteria. So sooner the environment is filled with fragments of the DNA coming from the donor cell. Now maybe in the same environment there is a recipient cell, the same cell from the same progeny, cell from the same generation. Now that recipient cell can sense the environment that is filled with so many DNA content, fragments of the DNA, so they can easily uptake any of those fragments inside the cell. Now after this uptake, that fragment can be recombined with the bacterial chromosome of that recipient cell. Now if it is recombined, then we know that as a new gene sequence is involved and incorporated into the recipient cell. And with the help of this new DNA which is incorporated here, they can have different features. The next one is the transduction. Transduction is a process where the DNA is transferred from one bacteria to the next with the help of virus. And the viruses that attack bacteria is known as bacteriophage virus. So this is the scenario. When a phage virus infects a bacteria, it injects its DNA inside the bacteria and then it starts breaking down the bacterial chromosome into fragments. Now the fragmentation occurred inside the donor cell. Now once they start packaging of their own genetic material inside the viral head or virus head particle, sometimes through mistake they take some part of the bacterial chromosome or bacterial DNA fragment inside any one of those phage head. Now the phage head, which carries the breakdown content of bacterial DNA, can be transferred because that phage can go and sit and inject its DNA into a recipient cell. Now the recipient cell will receive the copy of the other bacterial chromosome or bacterial DNA. Now that DNA could again be incorporated with the recipient cell, so they can have this way. So basically transformation is without any virus, transduction with the virus, and the process is similar. They only transfer the fragment of the bacterial DNA, but in transduction they use virus to do that. In transformation they just directly uptake that. But the third one is conjugation, which is completely different, which is a kind of a direct interaction between two bacteria, two adjacent bacteria. As you see here, they are involved with each other. with the help of a tunnel that is formed between them, known as the pili or F pili or pilus, you can say. Now with the help of this is a conjugation tube that is formed between the two adjacent cells, and through this tube they can transfer genetic content, they can transfer even plasmids from one bacteria to the other bacteria. This is a direct contact process. Now if we distinguish between the process of conjugation and transformation and step-by-step process, let's look at conjugation process at the beginning. We know in this process we have one donor, we have one recipient. The donor have a F pilus. F pilus means F stands for fertility here, and they also get a sense of plasmid known as F plasmid, a fertility plasmid, and this F plasmid containing bacteria always become the donor because they will donate this F plasmid to the recipient cell. Now those who carry this F plasmid, we call them kind of a male, okay, resembles like a male, and the recipient resembles like a female in this case, and this whole process kind of resembles like the sexual reproduction process, but it's not exactly. Now in this case, with the help of this S pilus, donor attaches with the recipient and they form a tube in the middle, and this tube is known as conjugation tube. They form this conjugation tube in the middle. After they form the tube, then they can easily start replicating this F plasmid with the help of rolling circle mechanism. With the help of this rolling circle replication, they start replicating, and they meanwhile they are transferring the content of this F plasmid through this conjugation tube to the adjacent bacterial cell. And then after this whole process is complete, both the cells will receive one copy of the F plasmid each, because F plasmid was earlier present in the donor, but now the recipient will get one F plasmid. So once it gets the F plasmid, it gets the characteristic of making this F pilus, and now it can again interact with some recipient and do the same. So this is the overview of conjugation. While in transformation, we have the fragments of the DNA which is generated after the death of a bacteria. Okay, so bacteria is dead, the cellular components are destroyed, and the DNA fragments are floating in the environment. So another adjacent bacteria comes in, it senses the environment that there are fragments outside, so there is a possibility that some of this fragment may contain any sort of antibiotic resistant gene. If it carries antibiotic resistant gene and somehow this recipient can take that fragment of the DNA containing antibiotic resistant gene, then this recipient bacteria can get the antibiotic resistance as well. That is the idea of transformation. Now in this case, they uptake the fragment of the DNA, and now it will be incorporated with the recipient cell. So after this uptake, they incorporate the fragment of the DNA into the cell with the help of recombination. Okay, they use this homologous recombination process to properly place this DNA in their genome. Okay, by this type of fashion they gather or acquire a lot of different features of producing different like enzymes, producing different sort of chemicals or enzymes which will fight against antibiotics, and also antibiotic resistance factors from different cells they can gather by this transformation process. The last one is the transduction, and if we look at the process in details, here we go. The donor bacteria where a bacteriophage infects, normal process of bacteriophage entry is the injection of phage genome inside the host bacteria. Then the host bacteria's DNA is degraded by the phage. So once the phage does that, after that phage DNA itself start replication of producing more phage genetic material. Once they produce so many phage genetic material, then they process those material and put them inside the capsid of the new progeny phage. During this packaging, this is known as the packaging. Okay, packaging of DNA, phage DNA inside the phage head. During this packaging, due to some mistakes and problems, sometimes they also put some of this bacterial DNA into the head of the phage. Now this is rare though, but it happens. Like for example, in this picture you will see 1 out of 5 phage particle receives the bacterial DNA. Now it transfer, once that bacteriophage go and transfer its content inside the other bacteria, which is here the recipient bacteria. You put it inside the recipient bacteria, inject that DNA inside. In that case, now this recipient bacteria receives the DNA content which should be phage content, but in this case that is the bacterial DNA content of the donor. In this fashion, a recipient bacteria receives a part of donor bacterial DNA. And in this sense, that DNA can be incorporated with the recipient bacteria genome. This incorporation may be complete, and what we get? New gene acquirement. So in all these processes we see that there are involvement of different processes, but all these processes are required mostly for the gathering and generation of antibiotic resistance properties by the bacteria. So that in a sense is the difference between conjugation, transformation and transduction. If you like this video, please hit the like button, subscribe to my channel and share this video with your friends.